Not yet. Is it going to be this semester or next? Next. Okay. Yeah.
So exciting.
Thank you. I'm pretty sure everyone's on board, but again, I have to talk to them all. But I think I have enough with this project and the next one I'm working on, which I'll touch on a little bit in this, that I'll have enough data for defense. Yeah.
Glad you could join us, Dr. Kinney. And I will forewarn you, we will start right at noon sharp because we have a hard finish at 1 o'clock.
Okay, that's completely fine.
Because now there's another.
Is there another class right after this? Okay.
How are you gonna do like 110?
110, yeah. So we have a couple minutes of leeway.
I just realized that camera in the corner was gone.
Yes. I think we first encountered camera issues during week 2 of the semester. We're now what, week 8?
Yeah, I love when the technology doesn't work very early on.
Hello, Dr. Scott.
That's the way. How you doing, bro?
Hey, doing good.
Wow. Well, it's okay with you, Dr. Scott. I'll let you
Okay, sure, I'll be happy.
introduce that part of it then.
This
Oh
is
yeah,
stuff that,
with the wound
I
infection.
think we got
stuff.
somebody lined up to get us some samples.
That'd be super cool just to look at those
I
isolates.
don't want to filter anything to see if any plastics have Vibrios.
That's true. Could do some microplastic work as well.
Good morning, Dr.
If you want to,
Bejarano.
I mean,
Yeah.
Sean
In our
has-
free time.
Yeah,
Work
my
on
free time.
dissertation.
Just another project.
So, Dr. Scott, we are making progress on our community engagement core. I sent out another update this morning.
So will you have any more write-ups coming in from your subs, or you'll be covering all the write-up for them?
No, they've got to send it in. So I've got a draft from Keith already and talked to Keith. And my biggest concern is just the latency grab. They're always the last minute. But we have spoken to everyone.
No, I saw that. And I'm gonna try to get that draft letter out to Heath from last time. So
Okay.
he's got something to go. I'll put the title in. So
That would be fantastic. Do you recall, and I can't find your email with the margins?
it's in that attachment that came with the outline.
Okay.
If you can't find it, I'll send it to you as I get back to the office.
I think our biggest issue will be keeping it down to 6 pages.
Yeah, well, that was why I was asking whether or not you're doing the write-up for the,
See,
I don't think we did a write-up for the
because
subs last,
I don't want to.
Well, so what are you referring to as the write-up?
like the technical write-up for the whole course.
Okay, yes, so we did
You're gonna
incorporate
cover
that in.
the other,
Yeah,
so all you're getting from them is 5 sketches,
Because
but
people
correct, yes.
facility stuff, all that.
Did they also have to submit text with them with proposal? Did they have to submit their statement of work for SAM?
Right.
And so I already have the draft of that with Heath because he's in, I don't know, halfway around the world.
Brian's in Jakarta. Oh, man, he is a world traveler.
Folks, we're going to wait another minute or two to see who else either comes online or shows up.
Can I make a very brief graduate student announcement?
Of course.
DSAC has a survey that hopefully has gone out through the listserv to everybody. It's completely anonymous. If you'd like to share a little bit about kind of our work and like the culture in your lab and stuff. And it's entirely anonymous and just being used at the college level. And then secondly, DSAC is hosting a tailgate that all students will impact the individual who wants to come for October the 29th. It's against Missouri. We will be in the fairgrounds, and there will be stuff posted to the listserv, to social media, and then as well as around our buildings. So yeah, if you'd like to come join us, hi everybody. We're trying to get a little more social events going on. That's all I got.
Thank you, Lacey.
That'll be in one of the cabooses.
No, they don't fund quite that much.
Okay.
We will be in the
I
fairgrounds.
didn't know if it was a thing sometimes. I didn't know if y'all were doing it
Well, I guess we should have asked the dean for a caboose.
there.
Missed opportunity.
That's good.
So Dr. Deco, as Ryan is one of your students, would you like to have the honor of introducing him
Sure.
today? Sure. And I will tell you that Dr. Scott, after you do the introduction, Dr. Scott has some information that really points to the relevancy of the work that you all are doing.
Well, today we have Ryan Leighton. Ryan joined us in 2019, just a few short years ago during the pandemic and so forth, and came here as a dissertation student. He wanted to work with plastics, biofilms, bacteria, and a whole number of other things. And I think Ryan's case illustrates what happens typically in dissertations. He planned everything out that he wanted to do. And the first thing his committee said, and I said, it's too much. This will take you 25 years. And so he narrowed it down, he, thought about things, narrowed it down, and then came up with a shorter plan. And then as he proceeded, he realized he had to modify each and every one of those steps, which is typical in dissertation research. You plan things perfectly and it doesn't work, you have to modify it. That's research. He's working on plastics and Vibrio bacteria, which are potential pathogens. And plastics are a very common pollutant, toxic component of our ecosystems that we may or may not see. We've heard of the great garbage patch the size of Texas and similar things in the oceans. Ryan is working with things we can't see, microscopic particles of plastic that may transport bacteria over distances. And how does this change as climate is changing? There's a lot of interactive steps to this, and it's coming out to be a very interesting story. So I want to introduce Ryan Leighton and some of his dissertation research, which he'll talk about and tell us about today. Ryan?
Thank you so much. You wanna
Yeah,
talk about-
just to put a little context on this, I got an email late yesterday afternoon from Dr. Mike Parsons, who's the director of the OHH Center for Ciguatera poisoning down in, unfortunately, Fort Myers. And he informed me they were having a huge outbreak of Vibrio vulnificus infections, wound infections. 4 deaths, 29 cases since the storm. And this year in Florida, they've had 11 confirmed deaths and 65 cases. Last year, they had 10 deaths and only 34 cases. And those 29 illnesses and 4 deaths were just in one county, right by Fort Myers. Anyway, what's most interesting, a recent paper came out from the University of Florida showing a 1% increase in sewage discharge, the nutrients in that, will increase the abundance of Vibrios by at least 100 to 1,000 times more than their present level. Carlin's research has shown the same sewage, not the exact same sewage, but nutrients from sewage as well, the work she and Sean are doing, turn on and transform the genes that switch from putting a flagella out to find a host to putting out biofilms, which stick to particles. And I assume that can also be people's legs walking around in floodwaters. So this is a wonderful opportunity. And Sean, I did get an email back from Mike who talked about it. But of course, they can also absorb the microplastics from, which is a perfect introduction to, and in fact, it may be playing a huge role in some of the exposure we're seeing. So
For sure.
the floor is yours, sir.
Cool.
Thank you for that introduction. And thank you everyone for coming. The title of my talk today is Vibrio Paleolithicus and Vibrio Vulnificus Colonization on Plastics, Influenced by Temperature and Strain Variability. So this is part of our work for the Centers for Oceans and Human Health and Climate Change Interactions, part of our microbiological core, and this is funded by the National Institute of Environmental Health Sciences. So for my presentation today, I'm going to go a little bit in the background of the problem of marine plastic pollution. As you can see from this picture, there's a lot of different types of plastics, and basically the implications of this for what I'm interested in with bacterial interactions. And then I'm going to talk about a brief overview of my project and methods, and then we'll go more in depth with the results. And finally, we'll wrap up with conclusions, my next steps of what I'm working on now, and what I would probably recommend for future research and monitoring, and then final takeaways. So with looking at all these different types of plastic, there's a lot of different types of plastic classes. So they're made of different combinations of hydrogen, carbon, usually. The ones I'm particularly interested in are low density polyethylene, polypropylene, and polystyrene. There are obviously a lot of different other plastic classes, but I want to draw your attention to specific gravity here. So when you're looking at the 0.91, 0.93 of low density polyethylene, and then polypropylene with 0.85, 0.83, so those are less dense than seawater, so they're going to float. So those are common ones you're going to see contaminating the marine environment. But I also wanted to look at polystyrene itself because it actually is more dense than seawater, so it can sink. So I wanted to look at different dynamics of how plastics basically just interacting in seawater. So we know with plastics, they can either be macroplastics, which are large pieces of plastic that enter the marine environment from different pollution sources, and then over time, they actually will degrade smaller and smaller into microplastics, and that's due to shear stress from waves, from currents, from UV light, from the sun. So when they become microplastics, I know there's a lot of attention in the news now because we're finding out microplastics are in human blood. Microplastics are going from mom to baby in utero. So while we're still trying to figure out what microplastics are doing to humans, we're uncovering more research of what they're actually doing in marine animals. So there's a lot of different effects that microplastics can have from different exposure pathways. And what they've seen is it really does affect growth, reproduction, and mortality in a lot of different marine species, which reduces their overall fitness. And that means it dampens our food security because if we have aquaculture species or wild caught species that are more affected by microplastics, our quality of our seafood decreases. Now, while here in the United States, we have a lot of different protein sources, but this is a huge issue in countries that rely on seafood as their main protein source. So it's only going to get worse in addition with climate change. So we have like almost this double trouble with microplastics causing problems in marine animals and then climate change exacerbating those effects. So while there's a lot of different exposure pathways, usually with ingestion that we're interested in, I'm particularly interested in microbial assemblages. So what I mean by that is bacteria that live on different plastic materials, and these are called biofilms. So what a biofilm is, you can see from the life cycle over here, basically you have planktonic free-floating cells in the marine environment. They sense a substrate of some sort, they attach that substrate, and they start secreting the slime, which is called extracellular polymeric substances. And this slime basically anchors the bacteria to a surface, and it protects them from the outside environment from different stressors, whether it's temperature, pH, or even as you're seeing from this figure, different protists or heterotrophs that are feeding on the bacteria, the ones basically at the lower layers are protected. So with this life cycle too, once they're become more mature biofilms, if the conditions are not right and they're basically stressed within the biofilm, they can detach and they repeat this life cycle all over. We're finding another surface basically to colonize. So commonly found biofilms in the marine environment are on ship hulls and on rocks. If you've seen probably slimy rocks, it might be algae, but it could also be different communities of microbes living on those rocks. But it is interesting with biofilms on ship hulls, and just a little interesting thing I found that with biofouling, so that's where the biofilms coating on a ship are actually eating away at the material on the ship. But the biofilms actually over time, if they build up on the ship hull, they're creating more weight on that ship and actually creating more drag on the ship, which is actually increasing the amount of fuel ships have to use, which is super interesting that you think of just like little small microbes, but if there's a ton of them, they can actually basically slow the ship down. So we're talking about biofilms, and now with essentially plastics, as I said, they degrade in the marine environment due to shear stress, due to UV light from the sun. They can become smaller. And this term called the plastosphere was coined about a decade ago. So researchers basically took plastic particles and they looked at what's living on these plastics. And they actually were pretty surprised to find a lot of different species, not only bacteria, but other different animals living on these plastics. So you've got all these communities of protozoans, phytoplankton, zooplankton, bacteria, all living close to each other and all interacting with each other. So the implication from that is if you just had bacteria that are free floating in the marine environment, maybe they're not as close together as if they are now, if they are attached to a surface like plastic. And that means that can facilitate them exchanging genetic material, whether it's resistant genes to antibiotics, whether it is pathogenic genes. So maybe the plastics could be enriching pathogens on these surfaces. So what that could also mean is if you have a plastic material that potentially has a lot of bacteria on it, what happens to the fish? They might mistakenly eat these particles, or they choose to eat them because they look like food particles. So they can become maybe increasingly more contaminated because they're eating these plastic contaminated particles. So one of those bacteria that I'm interested in as a genus is Vibrio. So there's a lot of different types of species of Vibrio, and they're gram-negative, curved rod-shaped cells. But looking at the plastosphere, the communities of bacteria that are living on them, a study was done that basically took two different samples from sediment and the open ocean, and they wanted to see what bacteria are living on them. And surprisingly, Vibrio was dominant on this polyethylene particle that came from sediment, and Vibrio was dominant from this open ocean particle of polypropylene. That doesn't mean every Vibrio in here is pathogenic. That just means there's a chance that there are pathogens living on these different plastics. There's a lot of different types of bacteria that they found, but Vibrio was one of the dominant genuses that were on these plastics. So in the natural environment, Vibrio were attaching, and as we've seen, they're hitching rides on zooplankton, on phytoplankton, they're living on animal carapaces, and that's in the natural marine environment. However, we've now introduced a new surface into the marine environment for them to colonize and live on. And what this picture is also showing is you could have a single species of Vibrio biofilm maybe living on a microplastic, but you could also have multi-species biofilms with Vibrio and other species of bacteria living close proximity to each other, which means higher chance of gene exchange, and maybe they're becoming more pathogenic. Also with microplastics, they can absorb different metals and different chemicals, and they're already made of toxic additives in addition to hydrocarbons, which could only possibly mean it's also turning on pathogenic genes because of these heavy metals and chemicals absorbed in the plastics. So we know that there's an increasing Vibrio threat to both not only marine animals, but also human health. So with the graphic on the left here, basically this is just showing that they did basically a modeling study, and they were looking at predicted changes of mortality of rare aquatic animals infected by bacterial disease in response to temperature. So the red dots are tropical species of fish that are raised in aquaculture. The blue triangles are subtropical species of fish in aquaculture. And this is essentially showing that when you increase the temperature, you're increasing the chance of mortality by bacterial diseases. So that's likely you're stressing out the animal first with increased temperature. And then because the temperature is increasing, it's also great environment for the bacteria to thrive in. So they're becoming more susceptible and more infected and leading to higher chances of death. And there was both a linear correlation with temperature in the tropical species and subtropical species. So, and this is not only Vibrio infections, this is also different types of bacteria that do affect aquatic animals. So we've got marine animals being affected, but we also have humans also being infected. So with foodborne infections reported in the United States, if you look at this relative rate here, if it's around 1, that means it's pretty constant of infections in the United States. If it goes below that, that means we're doing a pretty good job of treating or basically cleaning the bacteria from food so we don't become more contaminated or infected. However, if you look at all these different bacteria, it's like, okay, it's around 1, some are dropping, so we're doing a good job. But if you look at the green line here, over time, this is non-cholera Vibrio species, foodborne infections, it's also increasing. So we're having this increasing problem of Vibrio, not only in marine animals, but because we eat seafood, and if it's not properly cooked or we eat it raw, we also have a higher chance of becoming contaminated as well. So coupling all that together with this emerging threat of Vibrio and microplastics, I'm reiterating that in the natural environment, we had Vibrio that were, attaching to sand particles, attaching to algae, attaching to arthropods. But now we have this new surface in the marine environment that pathogens are attaching to. And I've kind of touched on a little bit of the interactions of the pathogens and plastics, but also something to know, and I've kind of mentioned with chemical additives turning on potential antibiotic resistant genes, but they are basically hitching a ride on this raft. They can travel much larger distances than if they were just free floating in the marine environment. And they could potentially become an invasive species too, if they hitch a ride and go somewhere that maybe they weren't commonly as found. And then you're thinking about the longer retention times in guts because of plastics that also increases the chance of contamination when an animal basically eats a particle. So again, I mentioned that there are different inputs of different plastics. They degrade, they're going to be become in uptake in marine food webs and also in aquaculture. And ultimately that means not only for the health of the fish is suffering, but also we're going to likely be more exposed because we eat the contaminated seafood. And then of course you couple that with the increased disease risk of warming waters and climate change. So it's an emerging issue that needs to be looked at more. And actually a study was done recently. If you look at the figure on the right, they basically took this oyster and they exposed it to virgin microplastic beads that had no biofilms on them. And then they exposed the oyster to microplastics that had just E. coli biofilms on them. And actually the oyster preferentially ate more of these microplastic beads that had biofilms on them. And what does that mean? So the oysters actually had more microplastic uptake. So they ate more of these, and it's probably because they are selective filter feeders. They're probably thinking these microplastics are food particles because of the E. coli living on the microplastics compared to nothing on them. So this obviously increased the respiration rate, increased mortality because now it's more stressed because it ate more particles. So we need to think about not just the particle itself and studying that, but also the interactions of other things that are living on these particles. Okay, so all that background information really inspired my research of my project. So this is an overview of what I've done in the laboratory setting. So I'll walk you through it. Basically, I took different strains of Vibrio of 2 species, Vibrio paleolithicus, Vibrio volnificus. I co-incubated it with one type of piece of plastic for 24 hours at different temperatures. After I grew the biofilm on this piece of plastic, I then wanted to assess how much biofilm biomass is on the plastic, and you can do that with crystal violet staining. That stains extracellular polymeric substances, which is the stuff that makes up the house of the biofilm, and live and dead cells in the biofilm. And I also wanted to weigh out the dry biomass to see basically how much, again, biomass is on these plastics. I then removed the biofilm from the plastic to get at the viable cell concentrations on these plastics, because I wanted to know if they're at a certain concentration, could that potentially, that size of plastic, be pathogenic if it were to be consumed, if it's like a high enough cell concentration living on the plastics. Then I took, also this is a disc coupon. So in this study, this is not microplastics. This is looking at just the overall trends of how the bacteria are interacting with different types of plastics. I do want to mention that. So then I took a slide coupon and then I incubated for 48 hours at 30 degrees. So 30 degrees was deemed to be a good temperature for 2 strains that had the heaviest biofilm formation that I saw. And then I extracted basically the extracellular polymeric substances, the EPS, to look at the extracellular DNA content, the carbohydrate content, and the protein content, because I wanted to see what exactly is the EPS made out of. And then lastly, I incubated all the different strains, again, at the same temperatures, 25, 30, 35 degrees for 24 hours. And I wanted to look at hydrophobicity. And basically, with hydrophobicity, you take these cells and you basically combine them with, in my case, I combine them with p-xylene, which is a hydrocarbon. And if they really like p-xylene, they attach more to it, that means they're more hydrophobic. And if you think about plastics, they're hydrocarbons and they're hydrophobic. So the more hydrophobic cells are, the more likely affinity they are going to attach to a hydrophobic surface, which is why I did a hydrophobicity assessment. So these are my different coupon types and characteristics of my Vibrio strains I used, and environmental conditions. So with a coupon type here, I chose borosilicate glass as my surface control to compare to plastics with the Vibrio's colonization. These are the 3 types of plastic, again, that I chose. Low density polyethylene, which is used in plastic bags, 6-pack rings, packaging film is a big one. I chose polypropylene, which is used in bottle caps, packaging film again, rope, and polystyrene, which is used in single-use plastic utensils and food containers. So for the Vibrio strains, I wanted to look at strain variability, which means I want to look at 3 different sources where they came from. So with both Vibrio paleolithicus and Vibrio volnificus, I chose a strain from each that came from humans. So one came from an enteric system from humans and Vibrio paleolithicus. One came from human blood with volnificus. And then one came from mollusk tissue, but one came from eels. And then I had 2 seawater isolates. So I wanted to compare where these are coming from, how they're interacting with plastics. And the presence of genes in the 16S rRNA type, those are just markers to show if they're more predisposed to infect humans or not. For environmental conditions, just wanted to reiterate, these are the 3 experiments I essentially did, the types of coupons I used, and the different temperatures. So again, I kept it around 25, 30, 35 degrees for 2 of them. And then that 30 degrees, because that was optimal for 2 strains growth, incubation time. And then shaking speed was an interesting one I had to play with. But I wanted because you can grow biofilms usually statically, not moving at all. They're just sitting there in the incubator. but I wanted to introduce shear stress because in the marine environment, it's constantly moving, you're having wave action. So I wanted to introduce that as well in my experiments. So when I'm talking about biofilms on plastics, you're like, can I even see that? Because these are microbes, but actually you can visually see it. So this was a slide coupon I incubated for 48 hours. I changed the media every 24 hours, but in total it's 48 hours. And you can see this gunk on the sides of it, and that's actually biofilm growth. So visually you can see that they are liking particular plastics. And this was the Vibrio Paralymicus human pathogenic strain, and it really liked low density polyethylene. And when I was talking about the crystal violet test, you can see here, the darker the purple there is, that's actually biofilm. And then compared to your controls. So this was my glass as my control substrate. This one was low density polyethylene. This row was polypropylene, and then polystyrene. So again, you're gonna have some innate background staining, but if you're comparing the control that didn't have any bacteria in it compared to the ones that had bacteria in it, you see how dark purple that is? It's a lot of bacteria, biofilm growth on these plastics in a 24-hour period. Okay, now we're gonna get into the fun stuff with the figures and the data. So I wanted to start really broad overall, and I just wanted to see overall colonization trends between glass, and I pulled all plastic together. So all low density polyethylene, polypropylene, and polystyrene, I essentially combined to just compare it to glass to see what's going on. So at a species level, with just looking at glass versus plastic, it looks like Vibrio paleomyticus is colonizing plastic. Oh, sorry, let me back up. The bars are cell densities or mean colony forming unit per coupon, and the dots are biofilm formation or biofilm biomass. So if you're just looking at the dots here with Vibrio paleomyticus, at each temperature level, so blue is 25, purple's 30, red is 35 degrees, they're higher than the glass ones. And their cell densities are also higher than glass. What's interesting is you would look at this like, okay, 30, 35 degrees. So at increased temperature, they're colonizing plastic more just by looking at this. But then you look at Vibrio vulnificus with glass versus plastic. At much lower temperatures, it likes to colonize plastic compared to higher. So you're already seeing different trends between the species of how they're colonizing plastic as a whole. I will point out there are very high standard deviation bars. Again, this is just looking at overall trends between glass and plastic. And these high standard deviation bars are just resemble very high biomass variability between substrate surfaces right now. Okay, so now I wanted to go a little further. So now I'm looking at, again, at a species level, both Vibrio paralymicus and Vibrio vulnificus, but now I've introduced the three types of plastics separately, rather than combining all the data together. So at a species level, Vibrio paralymicus had significantly greater, this was done by a two-way ANOVA test, on low density polyethylene and polypropylene compared to glass overall. And the alpha is basically showing here that it also has significantly greater cell densities as well on these two types of plastic compared to glass. For Vibrio vulnificus, it has significantly greater overall biofilm formation on low density polyethylene and polystyrene. So again, we're seeing differences in what species might prefer what plastic. And temperature actually had a negative effect, as we saw previously, that if you increase temperature, you have decreased biofilm biomass. And again, I have to mention this high standard deviation bars, because we're going further and further, are because of high variability between strains. Okay, so now this is everything put together for Vibrio paralymicus. It's the individual strains, individual plastics, looking at biofilm biomass and cell densities at different temperatures. So the human pathogenic strain, which is ATCC 17802, and the mollusk isolated strain ATCC 43996 had significantly greater overall biofilm formation on low density polyethylene, polypropylene, and polystyrene compared to glass. But what's super interesting here is you're looking at ATCC 17802. So this one strain did really well at 30 and 35 degrees compared to 25 degrees. But if you look at the other two strains, you're actually seeing, wait, these strains actually did better at 25 degrees. So this is just showing that this one strain greatly influenced the overall species biofilm variability. So what I'm getting at is strain variability and looking at individual strains rather than just as a species is super important, and especially in plastic colonization. So again, with the mollusk isolated strain, when you increase the temperature, so with this 30, 35 degrees, you decrease the biofilm formation. And the seawater strain, while it did colonize the plastic, it was not deemed to be significantly different than glass. So for Vibrio vulnificus, again, individual strains, individual plastic types, looking at cell densities and biofilms with the dots. So with both the human isolated strain and the eel isolated strain, when you increase the temperature, you decrease the biofilm formation. You can see that with the blue dots being higher, and then you see these purple and red dots are lower. What was pretty surprising is the human isolated strain of Vibrio vulnificus, the colonization wasn't deemed to be significantly different than glass. And I thought that was surprising because this was isolated from humans, so an already stressful environment. So it's already predisposed to form pretty or heavier biofilm formation. And I would have thought at higher temperatures on plastics. With the eel strain, it actually was found to be significantly greater overall biofilm formation on low density polyethylene and polystyrene compared to glass. And then the seawater strain was the most surprising to me. It did have significantly greater biofilm formation on low density polyethylene and polystyrene compared to glass. But the seawater strain had the greatest biofilm formation out of all the Vibrio vulnificus strains, which I thought was pretty surprising that you basically just, you have a seawater strain out there that's performing much greater than a human pathogenic strain. Again, with this Vibrio strain, it is, when it was genotyped, it did have clinical characteristics which might play a role in why it's having heavy biofilm formation. So with looking at so this is a summary essentially of all that information I just threw at you. So on the left, and this is looking at percent change of all the different types of plastic from the strains at different temperatures compared to glass. So on the left-hand side, it's biofilm biomass showing percent change, and then it's biofilm CFUs or cell densities showing percent change. And the biggest takeaway from this, if you see green, that means there's a much greater positive percent change biofilm biomass on these plastics at certain temperatures. If you see red, that means it actually formed better on glass in those instances than plastic. And if you see yellow, there was no change at all. So you can see basically if you're looking at these two, with it, there are two strains that mainly stand out to me that liked glass more. It's the seawater strain of Vibrio pamlicus and then our Vibrio vulnificus human strain, which actually seem to do much better in some cases on glass compared to low density polyethylene and polypropylene. But again, we look at polystyrene here, and actually it did better on polystyrene compared to glass. Okay, so I talked about staining, total biofilm biomass with crystal violet staining. I also wanted to look at the two parts of a biofilm mainly, which is cells living in the biofilm, and then the EPS, the extracellular polymeric substances. And the big takeaway from this is I basically extracted the cells and the EPS separately, and then I freeze dried them and weighed them to see weights. And I just want you to look at the mean total dry biomass per weight per coupon. So this is combining cells and EPS. And if you look at the plastics with their weight compared to glass for both of these, is actually found to be significantly higher dry biomass weight compared to glass with these plastics. Also, I compared it to my crystal violet test because I wanted to see, are they getting similar kind of numbers, because they should. And there was a moderately positive correlation. So that does strengthen the assumption that crystal violet is an effective method of estimating biofilm biomass on different surfaces. So I talked earlier, I wanted to see what were the extracellular polymeric substances made of. So I analyzed the protein, carbohydrate, and extracellular DNA content of the extracellular polymeric substances to see what are the concentration of these and what percent weight of the EPS are these different biochemical components. And the main structural component, it looks like for both species I tested, so these are the two strains again I mentioned that had the highest biofilm formation at 30 degrees. It looks like when you see from this blue bar, protein content was the highest for all three types of plastic for both strains. And that is actually pretty consistent in the literature too with Vibrio paralyticus. They found that the structural component was mainly protein, then followed by carbohydrate, and then extracellular DNA. But again, this is just for mature biofilms, so biofilms that have grown for longer periods of time. So this might be different when the cells are first attaching to plastics, but this is just for mature growth on plastics. And lastly, so I talked about the hydrophobicity test. So again, we have our three strains of Vibrio paralyticus, our three of Vibrio vulnificus, and I wanted to see how well they were doing adhering to p-xylene. And again, p-xylene is a hydrocarbon. And the reason I was doing this is because hydrocarbons are hydrophobic and plastics are hydrocarbons, so they're hydrophobic. So the more hydrophobic a cell is, the more affinity it's going to have to attach to a hydrophobic surface like plastics. So with these cutoffs here, at 30%, it's considered adherence to p-xylene. It's considered hydrophobic. So you can see all the strains here were above that, so they're all considered hydrophobic. And interestingly, at 25 degrees, especially for Vibrio vulnificus, it actually met this cutoff of being highly hydrophobic. Does that play a role in them basically having higher colonization at 25 degrees? It might, but I'm not sure. But this is just showing that they are hydrophobic and likely have an affinity to attach to plastics, which we've seen with the data. They all did attach to plastics, but some attached much better than others. Okay. So concluding all of that data, basically anything in microbiology, it's like, it depends. So different strain types of Vibrio paralyticus and vulnificus were found to pretty much colonize different types of plastics pretty quickly, like in a 24-hour period. And they had pretty high viable cell concentrations too. The human pathogenic strain of Vibrio paralyticus was found to have the highest biofilm information on low density polyethylene and polypropylene at higher temperatures. But that contrasts all the other paralyticus strains and vulnificus strains because they actually had higher colonization at lower temperature at 25 degrees. And I've actually seen in the literature too that that is for vulnificus, there are strains tested, they actually did better at lower temperatures as well. And I just think that is surprising to me, but there are things I'm starting to learn now about dispersal of biofilms that maybe that's not as shocking. Again, all the strains were hydrophobic at all temperatures tested. Proteins were the main component of both of these species biofilms with these two strains. And again, as I said, with anything with microbiology, everything depends. So this colonization on plastics, obviously it's very highly variable. It depends on the species. depends on the strain and the plastic type, especially under different temperatures. Okay, so what am I doing now? So I alluded to biofilm dispersal before. So we're just looking at this figure on the right here. I've mainly been looking at this biofilm maturation on plastics, but I'm interested in the bacteria leaving the biofilm or dispersing to colonize a new surface. And what I'm interested in is different input signals, so different environmental host variables that might overall affect Vibrio wanting to leave the biofilm and colonize another surface. And I'm looking at pH, temperature, so pH of like our human body, because it changes drastically in our digestive system, and human body temperature, which is 37 degrees, and nutrient availability as well does have an effect on biofilm dispersal. Because I'm thinking, if, let's say we ate a microplastic and it went through our system, our digestive system, the Vibrio survived, are they now leaving this microplastic to colonize our intestines? Because that's where the infections are found from Vibrio pneumococcal or vulnificus. It's usually the lower, the small intestine. So I'm also looking at dispersal coming from actual microplastics. Again, the study I did here was bigger pieces of plastic so I can handle them. But now I'm actually looking at microplastics to see what are the interactions after being exposed to different pHs and temperatures and nutrient availability. Okay. So my biggest thing is this double trouble of biofilms on plastics. We know plastics are a huge problem, obviously, in the marine environment, but we need to start thinking more of the animals or the bacteria that are living on these plastics too. It's more dynamic than just a particle. And I also wanted to mention, maybe we need to incorporate biofilms and plastics into forecast modeling, because right now we largely look at planktonic cell growth, but we don't really pay attention to biofilms as much, and definitely not with plastics, with our forecast modeling of predicting Vibrio risk of exposure to humans. And I've mentioned that too with include biofilms and monitoring. So our primary focus has been planktonic cell growth. Maybe we need to look more at biofilms too in those conditions as well. Because this is just a question I'm thinking, are these Vibrio bacteria basically hunkering down in these biofilms on plastics in colder weather because we don't have as much exposure, there's not as much Vibrio planktonic growth, but maybe they're in these biofilms in colder seawater temperatures, and then when conditions are right, they're leaving these biofilms and now they're resuming planktonic growth. Just a question. And again, we need more sampling efforts and collecting biofilm aggregates or substrates from seawater to just have a better data collection, and not just with the plastics I mentioned, but there's obviously all different types of materials too in seawater, and even like tire wear particles are a huge one too. That how are they interacting with those? And then screening and testing seafood for plastic particle contamination. So obviously seafood is screened for bacterial contamination, but I don't think there's any job being done on looking at plastic contamination in seafood at all and screening. So that's something that also needs to be considered. And then lastly, because these Vibrio really like to colonize plastic, are they potentially utilizing it? Are they degrading the plastics? Are they using it as nutrient sources? So that's also something that needs to be considered as well, because I saw in the literature another Vibrio species was found to actually utilize a type of plastic. I can't think of it right now, but so there could be potential that these different Vibrio species are using it as a nutrient source. And my final takeaway here is, if you've learned anything from this talk, you have plastics that enter the marine environment. As we know, they can get smaller and smaller through different environmental processes. You've got your free floating little Vibrio that are swimming around. They find this nice plastic raft that they basically start to colonize and live on. And now you've got this biofilm on this microplastic, and it can become a vector and maybe travel large distances, or fish preferentially, or accidentally eat it. So I'd like to wrap up by saying that this project was funded by the National Institute of Environmental Health Sciences, Center for Oceans and Human Health and Climate Change Interactions at the University of South Carolina. And I would especially like to thank the Baruch Institute for allowing Colin and I to sample seawater samples to get my environmental Vibrio Paleometicus isolate. I'd like to thank Carlin a lot for her expertise in Vibrio. I'd like to thank my undergraduate team, Addison Creech, Karishma Marichetti, and Gracie Anderson. They've done a lot of work and they've learned a lot. I'm really proud of them, seeing them grow in a lab environment. I'd like to thank Leon Zhang and Dr. Kai for their expertise in statistics and running the ANOVA analyses. And then, of course, I'd like to thank Dr. Deco and Dr. Norman for all their guidance and help with any kind of Vibrio question, no matter how dumb it is. So thank you. And if you have any questions, we do have time.
Questions?
Oh, there are questions in the chat.
Let's
Okay.
see what we have in the chat here.
The slide's not progressing. Oops.
Hey, this is Bonnie.
Hey.
I was having trouble.
Well,
I was having trouble with the chat, but it
at least
I did
heard
get it to start. Yes. No, I got it to continue. Because it was a very interesting talk. Thank you for sharing all of your research.
So how do you think your results would change if you use seasoned nanoparticles taken from the natural environment versus a non-seasoned plastic?
So good question. I probably would think they might actually have increased colonization because of the weathering process. So in the plastics, it's probably, if they're already naturally in the marine environment, you're probably creating cavities due to weathering. So that actually makes it a better home for biofilms to colonize potentially. And if in the marine environment, maybe there are already biofilms of different species on these plastics, which makes it actually easier for other species to colonize and attach those biofilms. So I would actually expect if you took a particle that already had a biofilm on it, or just a particle from the natural environment, I would assume it would have higher colonization just compared to a coupon I purchased from some biofilm company.
Incorporate some competition studies.
Mm-hmm. Yeah. Yeah.
Everyone that works with these huge issues that we have playing our environmental isolate, and they are completely, they're gonna say.
That's your Alexa.
They find out the word different, they pathogenicity is completely different. The two isolates use higher biofilm load, one is clinical and one is experimental.
The human pathogenic Paleometicus one was human clinical, and our VV155 was environmental, but it was genotyped to have clinical characteristics, right? Yes. Yeah.
It has some clinical, but
But it wasn't obviously from the environment, right?
that is compared to the environment. What we see, has been seen before that is in the literature, is like most of the videos do not have these pathogenic genes.
Mm-hmm.
It's more, no, yeah.
Yeah.
How do you think that that's gonna change? Because you have BB clinical one higher biofilm, BB environmental one higher.
Yeah, so the biofilm characteristics are really interesting. I guess in the environmental of colonizing plastic, I guess they might have a better predisposition to become more pathogenic because again, they're living on these plastic islands with all these other bacteria. Maybe they're exchanging information to them, make them potentially pathogenic. I don't know, but largely in the environment, it is non-pathogenic Vibrio. You have some that are.
Yeah, but I may be overthinking what I think is
Uh-oh.
in the environment, we don't have one species of another.
Mhm.
We have both.
Right.
Do you think that maybe gonna dominate in the biofilm in the plastic?
Oh, I see. Yeah, because of their biofilm, I'm honestly not sure. That'd be again something with a competition study, to see within a biofilm, and you can stain them differently. Is one species dominating over another, and then where in the biofilm are they located too, kind of thing.
But based on your data, we can say that we probably can have more PV in that kind of plastic.
Yeah, because of like the lower temperature, like in marine environment. Yeah.
And Ryan, just kind of piggybacking off of previous two comments, you mentioned they're hunkering down when they colonize plastic. And it's interesting that you see a majority of protein being produced in the EPS. And bacteria produce EPS as a metabolic excess product where it's carbon rich, or for a survival mode and where it's protein rich. You know, proteins are an expensive molecule for an organism to produce. Carbohydrates, not so much. It seems from your high protein amounts in the EPS that they are in fact getting ready to survive. They're improving their survival mode, whether they're going inside an oyster gut, a human, or just floating around being exposed to whatever in the marine environment. And that's very interesting in itself. They're predisposing themselves to better survival, maybe with biofilm formation. It's an active process rather than a passive one.
That's actually a really good point, because I didn't think about proteins being their secretion of just being metabolically expensive. So maybe they are almost evolving to be ready to be like stressed, kind of thing.
Yeah, Ryan, are you able from your biofilms to quantify the number of bacteria that might be on the particles?
Yes and no. So I can quantify with the previous slides I've shown viable cells,
Okay.
but that doesn't account for persister cells in the biofilm or other types that are metabolically inactive that can still be in biofilms. But with my method of detaching the biofilms and getting an estimation of cell concentration, you can do that.
So you think those are culturable but not viable, or viable?
There could
but
be
not-
some, yeah.
That's good, because if you think about it, John Weinstein's shown every oyster, the average level in oysters is 15 particles. So if you've got 100 bacteria per particle, that's going to put you way above the threshold for Vibrio illness. And if we can actually quantify the number of bacteria per particle, John and I just went up and met with the ISSC in early September. And in our new proposal, we're going to be doing a national survey with all the coastal states to not just know the numbers in South Carolina oysters, but oysters all over the U.S. so we can develop a universal exposure model, because they currently do not have one to accommodate the Vibrios. And I'm just going to mention this. Graduate students, we've also enlisted John Jacobs from Oxford, who is the world's best Vibrio modeler, in my opinion. He puts out the only forecast. And so John is going to be accommodating students to come up to Oxford to translate their data into his model. So anyway, I think your stuff is ready to go. And we need to talk, because I really think that's going to be a wonderful opportunity to actually improve the forecast. John is so excited about this, and I really think if we can pull off the national survey and then do this, we'll actually know how different the exposure levels are, how different are the microplastics in different regions.
Absolutely.
You know, I'm really wondering with the hurricane, is that a microplastic dump? Is that like an oil spill?
Could be.
I mean, from
Yeah,
a
from all
microplastic
the
point
stuff
of view.
on land, absolutely.
So, you know, what's going to happen? I mean, I wonder if this surge, probably too early to say microplastics yet, but
Yeah, coupled with the sewage as
Yeah.
well, like there could be all these interactions
Yeah.
happening.
I think adding the nutrients and the microplastics to the model are what we gotta do. So we can get accurate forecasts. But this is a wonderful piece of work you've done.
Thank you.
So our last two questions are from online, and since you mentioned the Oxford lab, Dr. Scott, welcome, AK. Glad
Oh,
you
good, we got
joined
AK
us
online.
today.
So with the educated guesses for preferential attachment, so they do seem to attach more to copepods and natural particles. So what we've seen, they're found in higher concentrations on natural particles, but they're still found also in plastics. It's just another surface we've introduced, but from recent studies, it still preferentially attaches to copepods and natural particles.
And then Bonnie asks, if I can scroll down.
So there are, yes. So with previous slides, they've, I know Zettler is a big researcher. I think she is in Norway. They've done a lot of work in looking at the different bacterial species on plastics. And there's another study that was talking about Vibrio acting as hitchhikers on plastics. So they've actually taken samples of environmental microplastics and found specific Vibrio species, not just Vibrio as a genus, but Vibrio volnificus and Pyramidicus on these plastics. So there are studies. Just gotta plug it in Google Scholar. I can't think of the titles off the top of my head. I know it's Zettler's a big one and Kirsten is a big one.
And Ryan is considering doing this for the second 5 years of his.
Well, we're trying to get more
I hope
funding
not.
out. Right, I've noticed something on that plastic slide you had where the Vibrios are the dominant species. Did you notice that the
The
seawater,
cyanus.
the cyanobacteria and formidium, which is the cyano that kills all the dogs, those were highly enriched with the Vibrios.
Yep.
I'd never noticed those
So
two.
algae, they've, I don't have it in this
Wow.
PowerPoint, but they're talking about also microplastics acting as vectors of HAPs
Yeah.
that are attaching. And if you think of like a giant, like the Pacific garbage patch, you probably have a lot of different stuff going on with HABs, bacteria. They're all interacting with each other.
I had never noticed that. I've
Yeah.
seen that slide before. I just happened to look. I said, wait a minute.
Yeah.
And that was in the seawater sample, which you showed. The seawater sample was highly, that's if I read your results right, the seawater samples showed a lot more variability in biomass
Mhm.
formation, right?
Yep.
So that makes kind of sense.
Yeah.
You know, that your data really confirmed why Vibrios are the dominant species up there.
Because yeah, they're apparently doing really well.
Yeah.
Any other questions?
I think that's it. How about a final round of? Good job, Brian. So next Wednesday, we have our own Lacey who will be presenting. And unfortunately, Lacey, I'll be joining remotely since I'll be somewhere on the West Coast next week. So we will look forward to your presentation then. So thank you all very much for coming. Thanks to you all online for joining us today. And we will see you next Wednesday.
Had to sit in the hall. I couldn't watch him in. I couldn't watch him and listen.